Professor Martha Muñoz of Yale University, who works on the evolution of thermal tolerance in a fascinating system of adaptive radiation, the Anolis lizards. Previous to Yale, she was at Virginia Tech, and she did her PhD at Harvard University.
Thank you so much, Aiden, for that introduction. Thank you all for the invitation and the opportunity to be here. I'm going to load up my talk. Okay, are you all able to see that?
Yes.
Excellent, excellent. Well, thank you again. I'm so delighted for this opportunity to share my research with you, and thank you for coming out on a Friday afternoon to spend some time with me. So at the broadest level, my research is motivated by a general observation in biology, which is that evolution proceeds unequally across the phenotype. By this, I mean that some aspects of the phenotype get to evolve very quickly, essentially in overdrive, whereas other aspects of the phenotype can remain inert or nearly so for millions of years. So to give an example, and these are real examples, when compared to other aspects of their morphology, lizard genitalia evolved so quickly so as to be deemed, and this is from an actual press release, as evolving at super speed. Just as impressive is that some aspects of the fish mandible can evolve up to 30 times faster than others. Now, keep in mind that these are integrated structures that need to work together for feeding, and yet some features can evolve much, much more rapidly than others. So why is this? Why does evolution sometimes get to zip along on the Autobahn with, traits evolving at warp speed? And why is it in other cases, like being in New York City at rush hour, with the brakes pumped on the entire process? What causes this to happen? Well, I approach this question by studying the many ways that organisms interact with their environments and determining how those interactions structure micro and macroevolutionary patterns of diversity. So to give an example of what I mean, imagine a tropical Anolis lizard perched on a tree somewhere in the Caribbean. Its ability to grow, maintain its homeostasis and reproduce are all dependent on its behavior, morphology, and physiology. For example, its ability to acquire prey or court mates or defend territories is limited by the thermal dependence of its performance, its hydric and metabolic economy, its behavioral preferences, and its morphological dimensions. And all of these features in turn interact in really complex ways. So my research centers around demystifying all of the ways that animals interact with their environments and determining how those interactions structure patterns of diversity. And I do so in large part by examining how organisms interact with temperature, because temperature really is important. And it's especially important for ectotherms like lizards. So in ectotherms, the ability to perform a task such as sprinting is contingent on body temperature such that it's optimized over a relatively narrow range of temperatures and then decreases at higher and lower temperatures until the animal is effectively immobilized. This curve has a lot of important features. For example, there are critical thermal limits, temperatures above and below which locomotion literally ceases. This curve has a maximum performance. It has a temperature at which performance is optimized, and there's a thermal range across which performance is maximal or nearly so. My approach is to dissect this curve, the thermal performance curve, into its component features and assess the variables shaping their evolution. And I do so in reptiles and amphibians quite generally, but today I'm going to be focusing in on one major lineage of reptiles that has centered prominently in my research, this being anoles. So Anolis lizards are a genus of lizards found throughout the tropical and subtropical regions of the Western Hemisphere. They are especially diverse in the Caribbean, where more than 150 species can be found, and there's more than 400 species total in the genus. And part of this incredible Caribbean diversity of anoles reflects their adaptive radiation that has been characterized by the evolution of microhabitat specialists termed ecomorphs, which have independently arisen on each of the major islands of the Greater Antilles. And anoles are, of course, these incredibly colorful and charismatic organisms to work with. They have these extensible throat fans termed dewlaps, which they use extensively in social communication and territorial defense, and they're just really fun to work with. But today, I'm going to be focusing on my preferred type of anole, the LBL, the little brown lizard. And in particular, I'm going to be focusing on Anolis cybodes and its close relatives from the island of Hispaniola, in particular, this lineage that we know as the cybotoid anoles. And I promise that what this lineage lacks in orange and blues and greens, it completely makes up for with physiological badassery. Okay, so what do I want to cover today? I'm going to have two major topics that I'd like to talk about. First, I'm going to zoom in on this cybotoid lineage of Anolis lizards from Hispaniola to unpack the role of thermoregulatory behavior in phenotypic evolution. And then based on the principles that we derive from those studies, we're going to zoom out to the entire lineage as a whole and revisit the adaptive radiation of anoles with some new perspective. And hopefully at the end, we can revisit classic ideas of the role of behavior in evolution with fresh perspective and maybe, discuss some emergent knowledge and avenues for future research. Okay, so the reason I focused on the cybotoid anoles from Hispaniola is because Hispaniola is a very special island. It's very topographically complex. You can be 3,000 meters above sea level and still be on a Caribbean island, but only if you do so on Hispaniola. Hispaniola is dissected into two major mountain chains, the Cordillera Central in the east and the Sierra de Bauruco in the west. When you're at low elevation in Hispaniola, you get that mesic broadleaf forest that you anticipate of the tropics, right? But as you go up in elevation on this island, and I mean really up in elevation, the habitat starts to seed and the forest starts to give way to this relative moonscape of monodominant pine forests surrounded by rocky outcrops and bunchgrass fields. And it's quite cold up there, very cold, in fact. You're in the Caribbean, but you can be in environments that are 20 degrees Celsius colder than near sea level. And from the perspective of a tiny little ectotherm like a lizard, that's quite dramatic. It's not uncommon to get frost at the highest elevations of Hispaniola, even in the summer, and it is quite chilly, which I rapidly discovered during my first expedition, for which I was woefully underprepared to the point that I was using suitcases or whatever I could get my hands on to thermoregulate in my own way using my suitcase. But I had excellent company on the summits of these mountains in the form of anoles. So in the western mountains, at the highest elevations, you can observe this species shown here, Anolis armori. And in the eastern mountains of the island, you can observe Anolis shrivi. And these two montane endemics are part of that cybotoid radiation of Anolis lizards from Hispaniola that collectively, as a group, span the gamut of available habitat on the island. Now, not all species are found everywhere. Some species, like Anolis longitivialis and Anolis whitemani, are restricted to the low elevations of the island. Straumi and Anolis marcanoi creep into mid-elevation about 1,000 meters. Anolis cybodes, this is a very special species, essentially is found island-wide, except at the highest elevations of the island, when it's replaced by Anolis armori in the western mountains and by Anolis shrivi in the eastern mountains. And my initial forays into this system were purely curiosity-based because these are tropical lizards. So my first question was just how, if at all, are these tropical lizards behaviorally or physiologically compensating for the really cold environments that they seem to occupy? And so what we did was, along with my incredible team of undergraduate researchers, we scoured the island for as many populations of these anoles as we could get our hands on, and we focused on two key physiological traits, the critical thermal minimum, CT min, and the critical thermal maximum, or CT max. These are the upper and lower temperatures that define the thermal limits of performance, essentially the temperatures at which the organism loses locomotor ability. And the way that we measure these traits is exactly what you see here. We systematically cool, or in this case, heat lizards, flip them onto their back and determine the temperature at which they lose their writing response. And it requires really sophisticated equipment like this heat lamp and a bucket. So it's very field friendly. Okay, so here are the data that we got. On this plot, elevation is on the x-axis and CT-min or cold tolerance is on the y-axis. Every point is a population, and the color of each point will tell you the species from which the data were gathered. And what we observed was a very strong clinal relationship between cold tolerance and elevation, such that those high elevation anoles were up to 7 degrees Celsius more cold tolerant than their low elevation counterparts. This result made intuitive sense because temperature progressively decreases with elevation. Let's do the same exercise, but now with heat tolerance. Same plot, elevation on the x-axis. Now heat tolerance is on the y-axis. And what we observed, interestingly enough, was that there was about half as much within population variation for heat tolerance as for cold tolerance, and absolutely no relationship whatsoever with elevation. Whether at sea level or 2.5 kilometers above sea level, CT max hovers somewhere between 38 and 40 degrees Celsius. So this urged us to maybe take a deeper look at the temperatures that organisms were actually experiencing in their habitats to see if we could unpack this pattern a little bit more. So what I did was visit 4 sites from near sea level to almost 2,500 meters, and I deployed a lot, that's the scientific term, a lot of thermal sensors. It was something like 25 thermal sensors in each habitat to record temperature during the day and during the night. I'm going to start with the nighttime temperatures first. So we're going to go from sundown to sun up on the x-axis, and the different color shapes are going to denote the temperatures we observed in each of these habitats. And what we found was a lot of thermal homogeneity at night, like a lot of thermal compression. Within any given site, temperatures varied by maybe 5 to 10 degrees Celsius. And importantly, very little, if any, of that thermal variation was shared across sites. Correspondingly, you can conceptualize a nighttime environment on this tropical mountain as being somewhat structured altitudinally into distinct thermal zones that go from relatively warm temperatures at low elevation to relatively cool temperatures at high elevation. Correspondingly, this clinal structuring of cold tolerance with elevation makes intuitive sense. Let's do the same exercise, but during the day. So now we're going from sun up to sundown on the x-axis. These are the same 4 sites. What we observed was a lot of thermal heterogeneity during the day, perhaps unsurprisingly. Now, maybe it doesn't get quite so cool at low elevation, and maybe it doesn't get quite so warm at high elevation, but overall, there's a lot of thermal variation at any given site, upwards of 40 degrees Celsius, and a lot, much if not most of that thermal variation is shared across elevation. Correspondingly, the thermal environment during the day on Hispaniola looks something much more like this. Now, if heat tolerance varied in a way that paralleled what we observed for cold tolerance, what we would have expected is more within population variation and a shallower relationship, but some relationship between heat tolerance and elevation. But instead, the data look more like this. Very little within population variation and absolutely no relationship whatsoever with regards to elevation. And it turns out that there was a piece of this equation that was missing. So does someone, some brave soul, want to unmute themselves and maybe remark an observation about something this Anolis cybotes is doing that seems a little unusual, perhaps?
Looks like he's sunbathing.
He is sunbathing. Are you able to observe how this lizard is elevating its hind limb off of the surface of the leaf? And if you peek in a little bit more, you can even see that it's curling its front toes off the surface of this leaf. It's essentially behaviorally modifying its interaction with the hot surface of the leaf. And it turns out that behavior was the piece of this equation that was missing. So even though the idea of behavioral thermal regulation might seem intuitive, perhaps even obvious to you and me, it's actually been formalized as a concept for less than a century. To put this into context, we discovered as a community the structure of DNA roughly a decade after we resolved the notion of thermal regulation. So it's actually relatively recent in the literature, and it's worth going into what we mean by thermal regulation. So let's take a bird's eye view of a thermal habitat. Maybe deep in the forest, it's a little too cool for lizards, and maybe out in the open, it's a little too hot for them. Maybe it's that flanking vegetation that is Goldilocks, optimally warm for lizards. Let's go ahead and color those optimally warm patches purple, because why not? And let's imagine that a lizard were to randomly shuttle between all of the available patches in its habitat. Such a thermal conforming lizard would indeed experience the highs, the lows, and the in-betweens available in its habitat. But if, in contrast, a lizard were simply to preferentially shuttle between the thermally optimal patches in its habitat, such a thermal regulator would actually only experience in practice a relatively narrow subset of the temperatures available in its habitat, to the point that from the perspective of such a thermal regulator, the environment might actually look something more like this. And that's exactly what these anoles are doing. These are body temperature data gathered from field active lizards from the same exact sites from which we gathered CT-min and CT-max data. And what you observe is that across about 2.5 kilometers of elevational difference, body temperature only drops by about 1.5 degrees. To put this into context, if these same anoles had been thermal conforming, body temperature should have dropped by something more like 10 degrees Celsius. So something's obviously going on with these anoles. And what they're doing is effectively transforming a daytime environment that could look something more like the situation on the left into an environment that across elevation, from the perspective of the temperatures actually exhibited by the lizards, looks something more like the situation on the right. They homogenize the thermal variation in their environments. But we can take this one step further still. So back in the 1940s, when this concept was being formalized, one of the architects of this idea, Charles Bogert, who's shown here below, gave it a bit more thought, and in 1949 published one of, in my opinion, one of the most prescient papers in our field. And he said, wait a minute, by thermoregulating, these organisms aren't just modifying, the temperatures they experience in their habitats, they're modifying natural selection. They're buffering themselves from selection. And he postulated that the features of the phenotype that could be behaviorally buffered from selection should evolve more slowly than the features that cannot so easily be buffered from selection. And in honor of his pioneering insight, we now know this as the Bogert effect, but you might also know it as behavioral inertia. And it turns out that's exactly what's going on in these anoles. Cold tolerance, a feature that's under selection when temperatures are coolest, those temperatures at night and in the winter become much more thermally homogeneous, and these lizards are diurnal in their behavior. So this restricts thermoregulatory opportunities to behaviorally buffer the cold. In contrast, the upper features of the performance curve can actually be buffered from selection through thermoregulatory behavior, and these lizards are, it turns out, incredibly precise thermoregulators. As a consequence, not only is cold tolerance faster evolving, but it's up to an order of magnitude faster evolving. So at this point, though, I was a little struck by the fact, like, why are these tropical lizards going through the trouble of adjusting their behavior for these very cold environments? Why not just adjust their physiology for the environments that they occupy? Maybe it's the case that heat tolerance simply can't evolve for reasons completely unrelated to behavioral thermoregulation. So what I did was gather every datum I could find about heat tolerance in the literature from squamate reptiles, and I discovered more than 25 degrees Celsius variation among species. When we zoom in on anoles, we recover more than 20 degrees Celsius variation. So it's not really the case that heat tolerance can't evolve. It certainly does. Rather, I think these tropical lizards are capitalizing on the performance benefits of a higher core temperature. So in general, when ectotherms perform a task, if they do so at a higher core temperature, up until a certain point, they are often able to do so at a higher maximal performance. Okay, so up until now, I think I've been beating the drum about the potential arresting force of behavior, specifically thermoregulatory behavior on physiological evolution. But if anything, if you go into the literature and explore, conceptual framework surrounding the role of evolution, of behavior in evolution, if anything, the opposite view, that behavior should drive evolution, might be embraced as the more classical perspective on the subject. And one of the chief proponents of this idea was Ernst Mayr, who spent a large portion of his career traveling around the tropics of the world and looking for mechanisms underlying the incredible biodiversity of birds that he observed. And he postulated that this diversity in no small part reflects behavior. Behavior allows organisms to exploit new resources in their environments, or even enter new environments altogether, in turn exposing them to selection and potentially driving evolution. Now, I don't want to suggest that behavioral drive and behavioral inertia are mutually exclusive ideas. They certainly aren't, but they have largely been studied independently. And I wanted to explore the possibility that they might not be so independent, because it struck me that this behavioral thermal regulation, which is imminently evident in these lizards, might involve some behavioral switch in resource use that might actually impinge on selection on other aspects of the phenotype. Like, what exactly are these lizards doing to so effectively thermoregulate? And does it have evolutionary consequences for other traits? So in order to do that, what we did was sort of really mechanistically pinpoint how lizards are thermoregulating. And we did that by visiting 4 sites, 2 low elevation sites, 1 each in the foothills of the major mountain chains, and 2 high elevation sites, 1 each, 1 in the Western Mountains and 1 in the Eastern Mountains. And in order to understand whether organisms are whether and how organisms are thermoregulating, you really need 3 pieces of data. The first is some understanding of the temperatures that are available to organisms in their habitat, like some null distribution of the temperatures organisms could theoretically exhibit. And we gather these operative environmental temperatures by deploying these copper lizard models that had thermal sensors embedded inside of them. And this is literally copper electroformed in the shape of an Anolis lizard. And the reason we do this is because copper heats up and cools off at a rate that's comparable to actual lizards. So it has a similar thermal inertia. We deployed a lot of these copper lizard models in each habitat in roughly equal numbers on boulders and tree trunks over the course of 6 days, during which time they were recording temperatures every 10 minutes. And we used boulders and tree trunks because the cybotoid anoles, this lineage of Hispaniolan thermoregulating anoles, are what we call trunk ground anoles or trunk ground lizards, meaning they spend most of their life within the bottom 1.5 meters of the ground. So tree trunks and boulders really are their most commonly utilized perches. At the same time that these sensors were in the habitats recording temperatures, we were out in the habitats also scouring them for field active lizards, catching them and measuring their body temperatures. So here's me and some of my former undergraduate students doing exactly that. This, of course, being the really fun part of the study. Then we brought a subset of those lizards back to the lab and exposed them to a laboratory heat gradient. And the idea here is we want some understanding of the thermal range that these lizards are theoretically targeting in the absence of thermal constraints. Like, what are they actually trying to achieve? And how accurately do they do so? So before I actually get to the data, I'm going to give you some impression of what the data look like. So what I've created here, I mean, these are going to be histograms. So these are frequency data. And on the x-axis is temperature. Now imagine that this is the distribution of temperatures available in a given habitat. If the mean and range of body temperatures tend to overlap cleanly with what's available in the environment, we would infer that this population of lizards probably thermoconforms. If in contrast, the mean and range of temperatures is quite different from that which is available in the environment, we would infer instead that those lizards are thermoregulators. And this is just a visual heuristic. We can put numbers behind this as well. Okay, so I'm going to start with the low elevation sites. Here are the temperatures I measured on boulders. Here are the temperatures I measured on tree trunks. Here are the lizard body temperatures. Does someone want to unmute and suggest whether these lizards are thermoregulators or thermoconformers in general? There's no wrong answers. I've heard them all.
Seems like the thermoconformers.
That's exactly right, Aiden. They're actually, they're not perfectly thermoconforming, but they're much more on the thermoconforming side of the spectrum than the thermoregulating side of the spectrum. And when we superimpose the preferred range, we see exactly why. And the reason is that these low elevation lizards are kind of in an optimally warm habitat. Like, their habitat is actually fairly pleasant most of the time. Or put differently, these lizards don't have to be very behaviorally precise to achieve temperatures within their preferred range. So these low elevation lizards are not precise thermoregulators because they really don't need to be. Let's do the same exercise at high elevation. Here are the temperatures I measured on boulders. Here are the temperatures we measured on tree trunks. Mind you, highly scientific finding here, trees are colder than boulders. Here are the lizard body temperatures. So Aiden, Thermoregulators or thermoconformers?
Thermoregulators?
Definitely thermoregulators. And when we superimpose the preferred range on here, we see just how precise these organisms are at thermoregulating, near perfect thermoregulators who are exceedingly nimble at targeting temperatures that are really rare in their habitats. If we zoom in on this narrow little sliver of the thermal environment that's actually warm enough for thermal regulation to be effective, this finding of tree trunks being cold suddenly becomes really salient. And the reason is that nearly 100% of the temperatures that I measured on tree trunks were simply too cold for thermal regulation to be effective. In contrast, boulders were 5 to 6 times more likely to be within lizards' preferred thermal range. So put differently, an enterprising high elevation anole can effectively thermoregulate simply by eschewing tree trunks and opting instead to perch on boulders. And that's precisely what these anoles are doing. At low elevation, these cybotoids exhibit the arboreal niche that is typical of anoles, and at high elevation, they've almost completely switched over to become boulder-dwelling or saxicolous. Now, why does this matter? Well, it matters because our entire understanding of the anolis adaptive radiation is predicated on the incredibly tight connection between structural microhabitat use and morphological specialization. So put differently, when anoles alter where in the habitat they perch, it's a fairly good inference that morphological evolution might follow. So our next step was simply to bring adult lizards back from the wild and measure some morphological traits that tend to shift in reptile lineages that go boulder dwelling from arboreal ancestors. And these often distill down to skull and limb dimensions. So what I'm going to show you here are residual plots showing, I'm going to start with head height. So for example, a relatively high value on this plot would indicate that that population of lizards has a relatively tall skull. And what we observed was that both in the western and the eastern mountains, those high elevation lizards have dorsoventrally compressed skulls. Our next step was to bring lizards from high and low elevation back into the lab, let them have their young and raise their young under common laboratory conditions to see if these same differences persisted in the laboratory rear generation, if this dorsoventral compression has a heritable basis, essentially. And indeed it does. These same differences persisted in the laboratory rear generation. These high elevation lizards have dorsoventrally compressed skulls. Now the value of this feature is pretty intuitive. So these lizards don't just perch on boulders for warmth, they defend them as territories, they retreat under them when threatened, and they sleep under them at night. And dorsoventral compression eases access into the crevices that these organisms now occupy. Likewise, we also detected reductions in the tibia and the femur, resulting in relatively shorter hind limbs in these high elevation anoles relative to their low elevation counterparts. And those same differences persisted in the laboratory rear generation. Now, there's many potential mechanisms that may be at play here, but one feature that does pop out is that greater limb parity. That means greater similarity in fore and hind limb dimensions is a feature generally associated with greater stability during climbing. And these lizards now spend their lives essentially scrabbling on large boulders and cliff faces. Okay, so what's going on here? So essentially, I think what's going on is that it's not just the case that behavioral drive and inertia are both true. I think that's already been evident. It's rather the case that not only can they both occur, they might actually be, fundamentally linked with each other, because the same behavior, this perch switch to boulders that can so effectively shield organisms from selection on physiology, does so at the expense of exposing them to selection and morphology. Essentially, there's really no free ride in evolution. And to put differently, it's a reminder that niches are multidimensional and that resource use often spans niche dimensions. So for example, when this lizard has made a behavioral decision to perch on this hot leaf, it hasn't just made a behavioral decision about the thermal environment that it occupies, it's made a whole slew of decisions. It's made a decision about the radiative and hydric environment that it occupies. This perch that it's chosen has a certain height, frictive coefficient, and surface area. It's likewise made a decision about the predators, competitors, and parasites that it encounters. And any behavioral decision that an organism makes can have multiple, and in this case, potentially even contrasting evolutionary outcomes on different aspects of the phenotype. Okay, so at this point in the work, I was very intrigued by this incredibly strong signature of the Bogart effect in these lizards from Hispaniola, but I really started to wonder, is this a cybatoid phenomenon? Is this a Hispaniolan anole phenomenon? Is this an island anole phenomenon? Or is this something that can characterize Anolis lizards in general? And the work I'm about to describe was led by a former undergraduate in my lab, Giancarlo Salazar. And what John did was collect every datum that had ever been recorded for body temperature, heat, and cold tolerance in anoles. And this really was a Herculean task. And he supplemented these data with freshly collected data of his own from mainland species from Colombia. And our general question here was, is this signature of the Bogart effect phylogenetically restricted or something that can generally characterize anoles? Like, at what phylogenetic scale does this pattern dissolve? And our first step in this process was really to assess whether the signature of thermal regulation differs between mainland and island anoles, because mainland and island anoles differ in many regards, and we thought that thermal regulation might be an additional axis of difference between species. So what you see here on the x-axis is mean annual temperature. This is meant to be a relatively coarse proxy for the average thermal conditions that different species of anoles experience. And on the y-axis is mean body temperature as measured directly in the field. Now, keep in mind that these are, behavioral data and inherently noisy, but what we observed is that overall, the signal of thermal regulation is much stronger in the islands than on the Latin American mainland. So body temperatures remain generally higher and more stable in island environments. In contrast, and I think this surprised me even more, there's a surprisingly tight connection between the local thermal environment and the observed body temperatures of mainland species of anoles. So even at this coarse scale, we do get a stronger signal of thermal regulation on islands. And this is exciting because it sets up the possibility that the Bogart effect, this restraining effect of thermal regulation on physiological evolution, might correspondingly be stronger on islands than on the mainland. And if so, we had a couple of hypotheses that we could test. First, if thermal regulation is resulting in the Bogart effect, then we would expect a slower rate of heat tolerance evolution on islands. We would expect that through this thermal regulation to a higher core temperature, organisms are evolving to a higher optimal value for heat tolerance. And if it really is behavioral thermal regulation that's sort of running the show here, then almost as a sanity check, we expect there to be no differences in cold tolerance between mainland and island systems. So what we did to compare the evolutionary dynamics of heat and cold tolerance across mainland and island contexts was fit a series of evolutionary models to the data and then determine which models best explained heat tolerance and cold tolerance evolution. The simplest of these models was a single rate Brownian motion model in which the differences between heat tolerance among species or cold tolerance among species is simply proportionate to branch length or time. A slightly more complex Brownian motion model allowed the rate of evolution to vary between mainland and island contexts. The other three models were all adaptive evolution models or Ornstein-Uhlenbeck models. The simplest of these assumed that there was a global phenotypic optimum for both mainland and island anoles for heat tolerance or cold tolerance. And the more complex model allowed the location of that phenotypic optimum to vary between island and mainland species. So in total, there were 5 models that varied in complexity, and we ran these across hundreds of stochastic character maps. And what we discovered was that the evolutionary dynamics definitely differ between cold and heat tolerance. For cold tolerance, a single rate Brownian motion model was sufficient to explain the phenotypic differences observed among species. You don't really need to invoke any different processes at play between mainland and island habitats. In contrast, the evolutionary dynamics of heat tolerance definitely differ between island and mainland anoles, both in the location of the phenotypic optimum and in the rate of evolution. The question, of course, being, does it vary in a way that's consistent with the Bogart effect being stronger in the islands? And if so, as a reminder, we would have expected a slower rate of evolution and a higher phenotypic optimum for heat tolerance. And that's precisely what we observed. Heat tolerance evolves 3 to 4 times more slowly in the island systems, and it evolves to a higher optimal trait value, about 4 degrees Celsius warmer on the islands than in the mainland. So this is interesting. We're recovering this signature of the Bogart effect being stronger in islands than on the mainland. But if you know anything about anoles, the idea that anoles, especially island anoles, could be slow evolving is somewhat weird, because if anything, as evidenced by their prolific adaptive radiation in the Caribbean, island anoles have been largely treated as a poster child of rapid evolution. And here we are recovering a signature of slower evolution in the islands than the mainland. And the mechanism for this prolific and rapid diversification on the islands is thought to be ecological opportunity. In the absence of their ancestral predators and competitors, these island lizards were able to essentially rapidly diversify to occupy all of the structural niches available on the islands. And yet we have a signature of slower evolution on the islands. So what gives? Well, these island and mainland lizards differ in several ways, but one of the most salient for this story relates to movement rates. Now, for context, my first expedition to work on anoles was in Costa Rica, a mainland habitat. And we spent weeks making behavioral recordings of lizards. And I was, this is my first experience with anoles ever. And I just had a camera, and honestly, I didn't need to move because the anoles spent most of their time doing exactly what this lizard is doing, which is not much at all, being as cryptic as possible and trying to remain unseen. And I just thought anoles didn't move around that much. And then I started working on island species and discovered, oh no, they spend most of their time running around their habitats, head bobbing, dewlapping, and being, quite frankly, as visually loud as possible. And this is the hallmark of ecological opportunity. This is that release from predation and competition at play. But greater movement rates don't just allow organisms greater mobility to exploit structural microhabitats, it also impacts thermal regulation, because the decision to behaviorally thermoregulate reflects a trade-off between its inherent benefits, for example, a performance advantage, against its inherent costs, many of which distill down to exposure to predators and competitors, which is precisely what these island anoles have been released from. When we put this all together, it leads us to the somewhat surprising conclusion that ecological opportunity, a feature almost universally associated with faster evolution, can actually slow it down by reducing the costs of thermal regulation. Okay, so at this point, we started to really wonder, well, how special are islands really for the Anolis adaptive radiation? Do these island environments provide any enhanced context for the Anolis adaptive radiation to unfold? And the work I'm about to talk about was led by a new and wonderful postdoc in my lab, Ed Burris. And what Ed did was compare rates of speciation across mainland and island species using hidden state models that he implemented in RevBase. And these approaches improve on previous approaches in two key ways. First, the use of a hidden state model reduces the likelihood of falsely attributing a rate shift to any given character. And second, the model explicitly accounts for background rate heterogeneity across the tree. And when Ed applied the model, what he detected was that there was no really a major or appreciable difference in the rate of speciation between island and mainland habitats. Now, he unpacked this a little bit more, and by focusing on the islands, wanted to see if speciation mode, in this case, in situ speciation versus speciation by dispersal, impacted rates of speciation on the islands, and found no signal whatsoever. Similarly, he also looked for island-specific effects. The four major islands of the Greater Antilles, Cuba, Hispaniola, Jamaica, and Puerto Rico, vary a lot in island size and in topographic complexity. But even still, he detected no major differences in speciation rate between any of the major islands. He unpacked this a little bit further still to explore rates of morphological evolution. And when he did so, he didn't find any major differences between mainland and island species, or even, depending on different speciation modes or among the major islands themselves. Now, when compared to their two putative sister lineages, this being caridophanid lizards or polychrys lizards, depending on your preferred phylogenetic hypothesis, we still recover anoles as more fast speciating, like faster speciating. So definitely there's something interesting going on in anolis lizards to the exclusion of their sister lineages. Now, anoles and their putative sister lineages have some key similarities and differences. They're both neotropical in their distribution. They're all semi-arboreal. They're very similar in many ways, but they also differ in some ways as well. Corydophanids and polycrysts both have dewlaps, although these are admittedly more robust in anoles, but they're lacking in adhesive toe pads. And these adhesive toe pads essentially function to create ecological opportunity by enhancing access to the arboreal niche. Now, obviously, the role of these toe pads in the anole diversification likely differed between the Latin American mainland and the Caribbean islands. Namely, on the mainland, we probably have a situation in which the toe pads facilitated cryptic arboreal diversification, and on the islands facilitated much less cryptic arboreal diversification. But importantly, the islands themselves don't seem to provide any enhanced context for this adaptive radiation to unfold. The patterns are exactly parallel between both of these major land masses. We were really intrigued by this idea that maybe these sticky toe pads, oh, actually, I should take a step back and say, these data that the mainland radiation is quite similar, actually, when you look at it, to the island radiation is part of a bigger emerging story from many different scientists and many different labs. So for many, many decades, much of what we knew about the Anolis adaptive radiation really was centered in the Caribbean islands, in large part because those simplified environments afforded a really amenable context for the study of these lizards in the absence of other features. But as more evidence over the past few decades starts to emerge, not just from our group, but from many, many groups, the picture is starting to become more complex. And it turns out that the more we look at these radiations between the mainland and the islands, the more parallels we're actually starting to find. We were, as one final note, intrigued by this potential of adhesive toe pads as representing some key innovation that might unlock diversification in response to the arboreal niche. And we capitalized on the repeated gains and losses of this feature in geckos to sort of explore whether there's a shared signal. And what we discovered is that pretty much in every case, in gecko sublineages possessing adhesive toe pads, speciation rates are uniformly higher than in the lineages lacking them. So this may hint at some generalizable principle and role for features unlocking arboreal diversification as being fundamental to enhance rates of speciation in squamate reptiles. Okay. So before wrapping up in the last few minutes I have left, I just want to give a hint to some of the work that we have ongoing in the lab, because some of it is quite similar and actually some of it is quite different. So we're still working with reptiles, but I'm also, we as a lab have expanded to include other systems, in particular plethodontic or lungless salamanders, and in particular the woodland salamanders from the genus Plethodon. And I've been fascinated by this group for a very long time for two reasons. One, they achieve peak biodiversity outside of the tropics, which is super weird for any herp lineage to do. So something super interesting off the bat is going on with these organisms. And secondly, they're lungless. And so they really are intimately connected to their hydric and thermal environments to ensure cutaneous respiration. So what we did was explore microclimate use in a sampling of species from these radiations. And what we've discovered is that microclimatic preferences for temperature and humidity seem to be structured along major phylogenetic breaks in this lineage, with species in the glutinosis group preferring relatively warmer and moister microclimates, and species in the cinereus group preferring relatively cooler and drier microclimates. Now keep in mind that these species will often co-occur, and we did the analysis. These don't reflect, the passive properties of organisms just behaviorally tracking what's available to them. We have hammered down the behavioral aspect of these preferences and demonstrated that wherever these species perch in their habitat reduces water loss rates more than would be expected given a random sampling of their habitat. So our next step is to sort of see whether and how these microclimatic differences translate into physiological differences, and essentially unpack the ecophysiological dimension of this radiation and sort of stitch it back together to determine how it contributes to spatial and phylogenetic patterns of diversity in this lineage. We're still looking at behavioral thermoregulation, but we're sort of expanding the phenomena under which we include it to explore other evolutionary phenomena, namely, for example, the evolution of live birth. And this work is centered on phrynosomatid lizards because this is a lineage in which viviparity or live birth has repeatedly arisen, making a really nice natural replicate to look for shared signatures of adaptation. And what we've discovered is that whenever these lizards go viviparous from an oviparous ancestor, their physiology uniformly becomes more cool adapted, but it doesn't just reflect the environment that they occupy. Using behavioral estimates, we've determined that even when viviparous species are in warm environments, they prefer cool temperatures. They really are cool adapted. And by merging these data with the metabolic theory of ecology, we've shown how cool adaptation reduces the energetic demands of pregnancy, which may have something to do with why these lizards are very cool adapted. Okay, we're still working on the Anolis adaptive radiation, of course. And like I said earlier, this is a story that has largely centered around exploring how structural microhabitat use contributes to morphological specialization and species diversity in the radiation. But this is really only part of the story. My lab centers around unpacking the physiological dimension of this radiation. In particular, these same anole lineages in the Caribbean have also diversified, as we talked about today, along the thermal macrohabitat axis with respect to elevation and in some cases latitude, and also with respect to thermal microhabitat along the sun shade axis. So work in my lab centers around putting the morphological and physiological dimensions together to get a more synthetic perspective of tempo and mode during the adaptive radiation of these anoles. Lastly, I just want to thank you all for being here and for giving me your attention on a Friday afternoon. And I hopefully will have time to take any questions you might have. Thank you.
Are there any questions? Well, there's one in the chat.
You were correct in your, I just saw your comments in the chat window, exactly correct observations.
Thank you. I think Monica has a question in the chat.
Here, how about I'll read it. What is the time frame of Anolis evolution? Do they have a sudden radiative evolution several million years ago and established in every niche since then, or do they still keep evolving at lightning speed with new species or ecomorphs popping out?
So some of this, I don't think I can share because it's my graduate student's paper that isn't submitted yet, but essentially physiological diversity seems to evolve closer to the present. That's some of the analyses that we're doing. Overall, the Anolis adaptive radiation is actually quite old. It's an old radiation. And the origin of some of those ecomorphs go back several million years. But you also get more recent evolution as well. And there's a lot of within ecomorph evolution, a lot of which is fairly recent as well.
Can I ask a question? I
would be delighted.
Okay. This is a little off topic, not 100% off topic. I've lived in Florida a lot, and when I was first working there in 2010, and I was at a lab that had a kind of mangroves around it and a moat, and we had a huge freeze, and all the iguanas fell in the moat and died. And we had the problem I was writing home about is my problem at work is floating dead iguanas. It's like living in a Carl Hassen movie. And, we had to hire, like people, it was horrible to get these things out of the ponds. But Florida has 3 species of invasive iguanas. And I'm curious if there's any thought about how these periodic freezes that call really large numbers of individuals are doing any kind of selection for thermotolerance within the populations that are invasive and or between the species that are there. There's iguana, and a couple of Tenosauruses, I think.
Yeah, so I actually, this is something that's has some studies behind it. So I can actually talk about this. So I'm going to talk about anoles first. So there is a great paper by Manuel Leal and Alex Gunderson. I believe it's 2010, 11, or 12 in AMNAT, documenting rapid evolution of cold tolerance or rapid divergence in cold tolerance in Anolis sagrii following their invasion of Florida, because it's an ancestrally Cuban and Bahamian species. And obviously it encounters colder temperatures in central and northern Florida than it does in its ancestral range. And they documented very rapid cold tolerance shifts in this species. A couple of months ago, a paper came out in Biology Letters led by James Stroud, showing that this phenomenon isn't just unique to Anolis lizards. In fact, when you look at the panoply of invasive squamates, and they had 5 different species that varied in how distantly related or not they were to each other, all of their cold tolerance shifted relative to their ancestral, what they exhibited ancestrally, and they all shifted downwards to a more cold tolerant mode of life. I don't know if these are definitely selective events, and evolution is happening in response to cold snaps. So for example, in 2017, there was a paper by Shane Campbell-Staton focused on Anolis carolinensis, largely in Texas, showing that in response to a polar vortex, there was a major selective event because of the lethality that induced. And he documented transcriptomic shifts in gene regulation in response to that persisted in the next generation. So definitely a lot going on with cold tolerance. There seem to be lots of reptiles that can, at least to some extent, rapidly modify their cold tolerance.
Thank you. Cool.
You're still going to get falling iguanas, though.
Oh, I know. Actually, I was really sad I grew up with carolinensis as the dominant anal in Miami, and it's too bad those guys could evolve their cold tolerance because they've been pretty much outcompeted. It's interesting you're finding about the movement, and I was trying to think back. I always thought of carolinensis as a pretty active animal, but I haven't watched them carefully in Puerto Rico or someplace like that to see that they move more.
Yeah, so carolinensis is an interesting case study because for a long time it was the only anole in the United States, right? So it kind of had unrestricted access to arboreal niche, and it's an arboreal species. It's what we call a trunk crown species. But in the absence of competitors, it will essentially expand its verticality on the tree trunk. It still uses high up perches, but it expands down closer to the ground, and therefore to the perspective of, about yay tall humans, we see them more. When sagrii is introduced, they still are there, but they just basically return to their ancestral niche of being higher up in the canopy. But they're still there. There's a great study by Yoel Stewart in 2014 documenting exactly that.
Wow, that's very interesting. So they're still there on top of the mangroves or somewhere.
They should be higher up in the canopy, which makes them harder to spot because they're much more cryptic. They're green, right? So that's really where they're supposed to be most of the time. I don't know. I know less about whether they reduce densities of carolinensis in some way. Potentially, I know less of that, but I do know that they're still there and they just move higher up in the canopy.
Yeah, that is very interesting, though, about about them expanding. So as many gazillions of years ago when I was a kid, they were on the ground. They're all over they're everywhere. And so I didn't know that about their ancestral habitat. And it seemed like there were plenty of birds still trying to eat them, but certainly they didn't have the competition. That's really interesting. Thank you so much.
My pleasure.
So we have time for one more question and then we'll move on to tea time. Does anyone? Oh.
Are any of the anoles parthenogenic? No, not that I know of. And they also are not viviparous, although some high elevation taxa will retain their eggs until a much later developmental stage, but they haven't made the full transition to viviparity. They can retain sperm, not the same as parthenogenesis, but sometimes you'll get surprised. You'll get surprises with them. They can retain sperm for very long periods of time.
All right, so I'm going to go make some tea, but feel free to stick around on this Zoom link and make sure to take a break, Martha.
I will. Thank you so much for being here. This was
Thank
really
you.
fun.
That was great.